I am really excited to talk to everyone about our research on pollinators of first food plants. First and foremost, no, I just need that minute. Oh, no, that's okay. Well, my name is Sarah Gardner. I'm a pollination ecologist from Pendleton, and Cheryl Shippentauer and I have been working on pollinator-related research for the last 5 years now, which is crazy. So I'm gonna just present a portion of that to you today. What I like to do is have, kind of a lively presentation. So if you do have questions in the moment, I'd love to address them as they come up. Oftentimes people, I'm just specifically citing myself, forget the questions at the end. So please interrupt me. I want this to be more of a discussion.
Notice the red bar.
The red bar is gone. Didn't work at first.
Well, I don't see the red bar.
I don't see the.
I hope they can see it.
Yeah, I hope everyone online can see. Right. So this is an overview of the discussion today. I'll be talking about the groups of pollinators that we have collected off of 4 first food plant species. Pollinator group is referred to as a guild, so I use those terms interchangeably. The 4 plant species that we sampled were Cous. I mean, I'm doing a really good job being super specific, so there's track. Okay, cool, thank you. Cous, wild celery, common Camas, and what people call either mountain huckleberry or big huckleberry. There's a couple common names. And then moving on to a project that we did last year, taking inventory of the entire bumblebee community across tribal land, comparing that species list to those that are found on first food. So it's interesting connection. We'll be discussing the pollinators of first foods first, which was research done between 2018 and 2020. Our main objectives were first, really finding out whether these plants actually require pollinators. Plants don't. So needed to determine that before we moved on to collecting insects that weren't necessarily doing the job we thought. And then if it was found that the pollinators were necessary for plant reproduction, what was the community that we found? We also did a monitoring of this community just to see if we could find any changes on big huckleberry in particular. So how these slides are going to be formatted is an overall discussion, and then I'm going to be talking about each plant species. The research was done within the ceded boundary of the reservation. Again, our understanding of the reproduction of these plants was unknown. So we had to determine whether or not these plants were selfing, which means that the pollen is spread between the anther and the stigma on the same flower. And that's usually done abiotically. Sometimes you do need a biotic vector, such as a pollinator, but oftentimes these plants can do the job themselves. Similarly, pollen can get transferred between flowers on the same plant. That's also considered selfing because there's no introduction of new genetic material. That can be done through incidental contact between these flowers, or sometimes it does require a biotic vector, such as the pollinator. and traditionally what we think of as pollination, which is the transfer of pollen between different plants, which can be done by a vector such as a bee. We wanted to determine whether or not these plants required pollinators, and it was unknown at the time for Cous, celery, and huckleberry what their reproduction system was. More research has been actually conducted on huckleberries since our study occurred. It is now well known that they do require pollinators. And then Camas, there was a paper that came out in 2014 that indicated that it is self-compatible, but it does require insect visitation for adequate. That could be even within the same flower or again in between. As many people have recognized, Lomatium blooms super early in the spring. So it can actually flower in March or April. You see celery right now in many areas around our valley. With that comes, super cold weather. It's not very hospitable for a lot of different insects. And so you'll have this selfing occur as a way that the plant can actually reproduce in these really unforgiving environments. So a study came out in 1982 noting that Cous was self-compatible, but they didn't know what extent. There was no actual data. So how we looked at the pollination system or the reproductive system is actually bagging inflorescences to prohibit access. So from left to right, we have the wild celery, Camas, Cous, and huckleberry. With celery, Camas, and huckleberry, they have these inflorescences on these big stalks or branches that you can actually cover in this white netting that prevents access to pollinators, but it doesn't change the internal temperature or humidity in the bag. So this will provide the same environmental conditions inside and outside the bag. With Cous, it was a little more tricky. They're kind of low to the ground. There's no stalk to put bag on. So we had to develop these cute little, what we refer to as bonnets, staked them on the ground around the plant. That prohibited those pollinators from accessing them. So basically what we were looking at is, exclusion of pollinators versus our control, which is open to pollinators. We wanted to look at number of flowers, and compare that to the number of fruits at the end of the season. So the flowers were covered once the inflorescence started to form, but before they actually bloomed. And then the exclosures remained until fruit set, so we could count the number of fruits. And, our results are again going to be presented in advance. Start with Cous. I'll just go through this quickly because it's been on each slide. What we found with our Cous prohibited from pollinators is not even a 1% reproduction. So open to pollinator services, there was about a 13% production rate. That's obviously very different. There was no significance in the number of flowers that we counted between our treatment and control. There was a significance in the amount of fruit indicating pollinators. And these trends continue with wild celery. Pollinator or plants that were excluded from pollinators had 0.3% reproduction. Open had about 21.5%. With Camas, this is actually a pretty big discrepancy. Plants without pollinator access had 3.5% reproduction rate. Remember, Camas had that self-compatibility element, so that could be in play even, noting that 3.5% was more than the other plants showed. But then it's 47.2. And huckleberry, similar to Camas, had 44.7%. Once we established that, every one of these 4 plants needed pollinators, well, what are they? What are visiting these plants? We're especially interested in the early season. Well, anything. What could be visiting these Lomatium early in the spring? Then we wanted to look at whether or not the pollinator community maintained consistency across multiple samples. We did that strictly on our. What we sampled was for each plant species, we selected 4 different sites. They were geographically quite separate so that we wouldn't be sampling from the same pollinator community. Most bees are able to fly, most medium to larger body bees are able to fly 500, 1,000. Some bees fly 30 meters. So we found populations that were typically over a mile apart, sometimes 10 miles. So we weren't considering, that we were sampling from the same group. How we did our survey is that we would observe pollinator actually collecting pollen nectar. A lot of insects will visit plants and rob them of either pollen and nectar in a bunch of different ways. But we wanted to make sure that these visitors were actually doing the job. We collected at a single site every day, and that's because there is a change in the community over time. So across the day, you'll have like early morning bees, like the bees that get revved up with coffee and everything. And then you could even get crepuscular bees, the bees that come out at dusk. So we wanted to stand for the whole community. The specimens that we collected from these plants were then dispatched and then taken to our lab for identification. Those samples are actually all out, so we can look at. Much like the reproduction study, I'm now going to be talking about each plant. Starting with Cous, we collected 121 specimens. 60% of these specimens were small ground nesting solitary bees in a single genus. That genus is Andrena. They're also known as the minor bees. You can see on the x-axis, there's labels for all of the other different groups that we collected, including sweat bees and the family Halictidae, some apidae, which are a Cous or 2. But for the most part, what really was found in abundance are these small-bodied Andrena. The 2 contenders that we found that visited with highest frequency are Andrena angustatarsata and Andrena W. scripta. So that could, angustatarsata was 28% of all the insects that we collected, and W. scripta was 15%. You can see it. Angustatarsata is on the left, W. scripta on the right. And in the center image, I was actually able to capture Angustatarsata, Forgione Cous. It's hard because they're really, they just like fly away very quickly. And you can see, those that are familiar with Cous, how small that flower head is. So now you know how big those bees are. They're quite small. With wild celery, we found similar patterns where we had mostly small solitary ground nesting bees. We also had a little bit higher abundance of small-bodied sweat bees. So these are actually primitively eusocial bees where you have a queen in the early spring. She'll start a colony, and then eventually when she passes away, her unreproductive children become reproductive and they start building the colony out. Very cool how they have this primitive social structure. And again, on the x-axis, you can see the other groups that we have collected. Not much diversity aside from the end. Once again, Andrena angustatarsata was collected in abundance. But the one that was found more commonly was Andrena melanocroa. which is the rose minor bee. They're very beautiful under a microscope. They have teal tinge in the light. And then again, the small sweat bees, which are indicated in the image on the right, they're about the same size as the Andrena plant. With Camas, we found 3 different trends. So we did continue to collect the Andrena bee. However, the primary group found in our collections were bees in the genus Bombus. Those are the bumblebees. We also found sweat bees, a group, the Beecology, those are the mason and leafcutter bees, and I'll talk about that in more detail in just a moment. And a few flowers. Most abundant was species was Andrena nigrocerulea. Iridescent bee, they're called the black and blue minor bee, and it's represented in the middle image. And we had 2 bumblebee species primarily. We had Bombus mixtus, or the fuzzy horn bee. They're called fuzzy horns because the males actually have on their antenna. And then we have on the right Bombus vancouverensis, the black notch bee. You can see on her thorax, she actually has a bee that comes down that meets with her abdomen, and that's why they call them the black notch. And those are found again in abundance compared to sex. With vaccinium, we did a multi-year study. So I'm going to show all of the years, one after the other, just to look at the differences, changes in the queen composition. Found that almost 60% of bees were bumblebees for 2018, along with some, sawflies, beetles, some flower flies, and wasps. 2019, again, dominated by bumblebees, some mason cutter bees, some wasps again. 2020 found nothing but bees, no beetles, no flies, no wasps. We found Andrena, all these bumblebees, some sweat bees, and it looks like 1 osmia probably. So, across all 3 years, 2 predominant bumblebee species were Bombus mixtus and Vancouverensis. But just to go over discussion, these plants do require pollinators. Very little reproduction was found when pollinators were excluded. Lomatium, they were primarily Andrena for both species of Lomatium. There are research included in 2020. This paper came out looking at the different species of Lomatium, and they also found that Andrena was predominantly collected. And Jim Kane was on my you see it? Yes, it's the minor bee. For the Camas, we found these 2 species of bumblebee and Andrena nigrocerulea. However, we also found Osmia lignaria, the blue orchard bee, pretty high density. Why that's important is that you can actually, it's a native solitary bee. You can actually purchase them. So if there are areas of Camas that are experiencing low reproduction rates for whatever reason, about it. Short term, work through whatever is happening and maintain this reproduction, putting these insects out on the landscape by pollination. It's very cool. And then again, we found the 2 species of bumblebee on vaccinium consistently over the 3 years. which were the same as what we found on the Camas. So Carol and I sat down and we were super excited about all these bumblebees that we collected. We weren't sure, how does this fit into the greater community of these bumblebees? We wanted to put it in context. So last year, we did a full bumblebee inventory. How these bees fit into the greater community. And in addition to that, we were also looking to potentially detect rare species. We have two in Oregon that are potentially going to be federally listed. That's Bombus occidentalis, the western bumblebee, and it's bee, Bombus nuclei. One of our other objectives was to look at areas of greatest diversity across the reservation, so that in the future, these hotspots, if you will, a bunch of different. And we decided to sample the bumbles because of their importance to first foods, but also they are easy to identify. There are resources out there that assist in the identification of these organisms using photographic rather than sample specimens. And that's interesting because you can then detect these rare, potentially federally listed organisms without having to batch them. You keep them on the landscape. And then bumblebees are unique. They're eusocial, an annual nest. And we were also curious to see if this we selected 6 geographically separate study locations. Again, we wanted to make sure we weren't sampling from the same group. The non-lethal protocols were borrowed from the Xerces Society. So how they have their large citizen science forward is that they pull these specimens down, thoroughly image them, submit the identifications to the Xerces Society, and they identify these bugs. And it's very cool. But then instead of us sending our data to Xerces, did the identification. What's really cool about this is, you know, it also associated with the plant host. So you're collecting off of these plants. You can say, I collected this Bombus sucklei off of this aster. And so then you also have this host. At each of these 6 locations, we found a suitable hectare plot. We would do a full bumblebee sampling for an hour and then do a habitat survey, which was essentially identifying all the flowering plants in that hectare. And each location was sampled at least 3 times. What we found were 11 Bombus species. They were collected from 10 plant hosts out of the 49 blooming plants that we identified, which is very cool. Most commonly collected was our Bombus vancouverensis. Is something that I wasn't necessarily expecting, but I'm not super surprised. It was found at 3 of the 6 sites, and it made up almost 30% of our plant. So the bumblebees that we sampled are all the center of this image. Surrounding that are the 10 species, host species of plant that we. These host plant records, it's really cool that you can actually say with confidence, bees prefer this plant. Highest visitation rate was found on larkspur, field ears, snowbird. Also, again, wanted to look at the diversity hotspot, finding across the areas that we sampled, where are we getting our species? What's really interesting is that last year's spring sucked. So cold and so wet until July. So I don't think this is necessarily an accurate representation of what the diversity could be if we did a multi-year study. This is a good snapshot. We sampled close to the native plant nursery here in Michigan. 2 species. Those 2 species don't overlap. They were found at no other site. And those species are known to be found in areas with invasive plants and disturbance. Tracks. There's one spot that had, even though there was a bunch of floral resources available, there were no bees collected. And I think that was because of the year that we collected. One area had just 1 cuckoo bee. I think it was looking for other bees to parasitize. But then at our higher elevation, more natural sites, lots of orbs, we found 5 species at each of these sites. They weren't the same species though. We had 60% similarity when you compare Indian Lake and Telephone Ridge. 2% similarity when you compare rainwater and Telephone Ridge. 40% similarity when you compare Indian Lake and rainwater. So there is a change in the community composition. However, the richness or the number of different species, same.
All different elevations.
In terms of when we could collect, yes, but there are certain bees in a subgenus that are only found at elevation. These other ones you can find between 1,000 feet and yard. We did not detect any rare species. And it's not that I think that we don't have any out there. I think those are crappy to do. So it would be really cool to be able to monitor these over time. We also found Bombus on insularis at 2 of our sites. That's our cuckoo bee of 4 of our bumblebee species that we collected. And no bumblebee nests were found to our. How do we keep these organisms abundant, plentiful for our first foods reproduction? And also we just love to see them. Well, maintaining or establishing native floral diversity in more urban areas, such as near the native pool. They do go to invasive plants. That's totally, like, that has been documented heavily. But invasive plants are like cheeseburgers. And the native plants are like whatever option is. So keeping them healthy with these native plants is then you can target plant those 3 plant species that we've identified as good hosts. So the snowberry, Anthea, just to start. We found a bunch of ground nesting bees, so protecting their habitat from ground disturbance, soil compaction, too much grazing, or changing from a wild area to agriculture. Maybe you can do that, but maintain some habitat. Using chemical free pest and weed controls or mating disruptions, that type of thing, rather than chemicals. And then discrete application of range and forestry management, such as prescribed fire. So fire can actually provide habitat by opening up their ground. You don't want to burn the whole thing because you don't have floral resources, but you then have nice little interconnected areas of habitat and flowers. And thinning of the forest to open canopy for huckleberry will also then provide areas for other forest. Yeah, this gets me all jazzed up. I want to do more work. I want Cheryl to do more. This kind of inspired us to ask the question, what what's the rest of the community doing? Now we have this subsample of bumblebees. What are the flowers that the rest of the community are visiting? What flowers are the first food pollinators visiting when they're not visiting first foods? Finding these networks, these connections. And also, you know, when I was out sampling for bumblebees, I found cool cuckoo bees. I found these weird panergines. I just wanted to know what's out there. So more selfishly for me. Thank you for coming. I, again, just want to shout out Cheryl. She's amazing. She's to work with. Gordy, who is the program manager for the Untitled Forestry, made me check in on days when I go out in the field to make sure I wasn't eaten by a bear. And Lindsay Kiona was clutch with finding sites at rainwater. She knows that place. I'd also like to thank Colin. It's really ambitious. And yeah. And I talked a lot, so I don't know if we have question time, probably not, but we can maybe walk out in the field.
We do have a little bit of time, so if you want to discuss a little bit, we also have a poll for you guys just to kind of structure some of
There's time, can we ask questions?
the.
For people,
Honeybee.
you know, how does that affect native species?
There has been more and more research that has come out to say, yes, most definitely. And that's not necessarily composition effects, but it's disease vectoring. So these organisms are visiting the same thing, and they aren't using this new start. So honeybees are vectoring all kinds of different pathogens that native bees haven't been listened to, so they're dying. Something that is now being managed by the Forest Service by restricting any honeybees from being placed on their or major conservative or target areas where honeybees can be placed where they know not going to interrupt the function. Honeybees are agricultural animals. They should be managed. They is with to be sold in person. Let wild sheep trade with our bighorns.
Sarah did a really good presentation on bees and flies and why. Introduction to isolated little home credits. Together. Myself. Dogs. Bee identification, actually finding nests and surveying, is that something?
Oh, yeah.
Getting the puppies out?
Yes. So there is an organization out of Western Oregon called Detection Dogs. So there is a wildlife biologist. She has been working with them to locate bumblebee nests and used by the training dog. But used to hone in on so that those dogs can go out. Because I mean, bumblebees use oftentimes road burrows, abandoned road burrows, little cracks in the soil that you can barely see with the naked eye. They're the same 3-foot patch for. So using these dogs, it's potentially another tool. What I like to do is I have an otoscope which is a thing that you can use to like the camera on a string. And just like looking at, it's got a little light on the end. And I like to feed that down into different cavities just to see if there's anything.
If I'm seeing a bumblebee, how far has he, what is his range? And where is he probably coming? Is there a nest near?
Yes. So the workers are foraging for pollen and nectar back to their nest. Their range ultimately depends what forage is out there. Interconnectedness, habitat, but typically it's about a mile.
Now, these bees I'm seeing are large.
Yes.
Did you see any of that kind of bumblebee?
So the queens, when they first emerge in the spring, because they hibernate over winter. When they first emerge from the spring, those are the ones that you see there. Those are all queen. There's the life cycle of the bumblebee is really cool. Where queens are hatched in the fall. And they mate, find a place to hibernate, and come out in the spring. So they often hibernate in say grass tussock or part of a. So they stay there until the spring and then they emerge. So those are the large bodied that you see. We'll start seeing workers in. So, but those queen bees are going to go forage themselves, but also to pick up nutrient start their. So once they get full hatch of brood to emerge, it'll take about 21 days. They never leave that nest again. They have their workers go out and collect.
They just lived the 1 year.
They just live the 1 year. Yep, it's an annual cycle. So it is kind of bittersweet, but then you get to see those big queens back out. Why? Might be. Yeah, you're like, well, why did the bees get bigger all over? That's the new queen gets going out. And their job is just. So if you do see those are the.
Well, I have a nest on my place then.
Well, if we don't have any more burning questions, we do have a field exercise to kind of get us into the group of our next one. Thank you so much, Sarah. Do you have any final words to kind of wrap up this a bit?
No, I can tell for 8 hours. So I could probably stop, but this is great. Thank you, guys.
You.
And we'll have a chance to discuss a little bit later. Too. So we're gonna start out there. So we have, do you want to introduce yourself?
Sure. Hey, everybody, I'm Brian Endres. I'm from just over the hill in LeGrand, Oregon State University. And also have the lovely opportunity to Cheryl. So, and tell her about it.
So we're going to go out and we're going to do some sampling, just like 10 minutes of each. And for virtual participants, we're going to throw on a video about what we've got going on in the road.
That's hard.
Video?
No,